Alright class, please look at page 1, slide 1 yang mind map ini. Sebelum ni ikut old syllabus kami memang ada anaerobic respiration. Tapi sekarang dia dah keluarkan anaerobic respiration. I think it will be better if you can replace this with fermentation sahaja. So tak payahlah tulis anaerobic respiration. Like what I explained to you the other day, anaerobic dengan fermentation memang lain. Dia tak sama. So this one, just put it as fermentation. Okay? Sudah?
Sudah, Miss.
Right. And then for your slide 4, muka surat yang sama, slide keempat untuk comparison tu, ini boleh kekalkan lah an example of anaerobic respiration, tapi you kena tahu fermentation dengan anaerobic memang tak sama. So maybe it will be better you stick with fermentation. Okay, ikut je fermentation. Kami memang tak belajar anaerobic. So, dah?
Sudah, Miss.
Now, page 2, slide 1. Muka surat 2, slide pertama, dekat jadual ini, Miss ada bagi anaerobic respiration. Ini tak salah, tapi you slash tambah fermentation. Okay? Tambah juga fermentation. Sebab I takut you nanti keliru. Ini anaerobic ke fermentation? Fermentation, dia melibatkan glycolysis sahaja. So, dia berlaku dekat cytoplasm sahaja. Kita akan belajar nanti. Hanya kena tambah ini fermentation. Sudah?
Sudah, Miss.
So, ini je lah yang saya nak betulkan. Sebab takut nanti masa exam you salah tulis ke, better keep it as fermentation. Alright? Instead of cakap anaerobic, lagi tepat kalau incomplete oxidation, itu memang fermentation. So, that's it for the correction part. And now, let's just go through ETC and chemiosmosis again. Okay, kita belajar semalam. So I will go through everything again. So let's go through all steps in aerobic respiration, and then we will go through oxidative phosphorylation again. Right. You learn that for aerobic respiration, kita ada 4 stages. Tapi the main stages memang 3 lah. Sebab kadang-kadang dia gabung link reaction dengan Krebs cycle. This one. Link reaction sometimes is considered as part of Krebs cycle. So jangan rasa confused kalau you baca apa-apa buku ada yang bagi tahu 3 stages saja. Itu pun betul. Okay? Tapi in our syllabus ada yang cakap 4 stages, but usually if refer to Campbell. Okay? Kalau Campbell, dia memang refer link reaction and citric acid cycle as one stage. Kiranya 3 stages tu lagi tepat. Okay, 3 stages, glycolysis, and then link reaction together with Krebs cycle. And the last stage, oxidative phosphorylation yang merangkumi electron transport chain dengan chemiosmosis. 2. I think you can write 3 stages atau 4 stages, I pun rasa betul. Okay, that's it for this part. And then when we look at the 4 stages or the 3 stages over here, let's go through these 3 stages together. We start with glycolysis again. I tak go through setiap compound tu, tapi kita just overview. Okay, overview the whole concept of, I dah salah copy ke? Yeah, it's okay. The whole, nanti ya. This one. Terlalu banyak pula. So we say the 4 stages or the 3 stages, mana-mana pun betul. Now let's start with glycolysis in the cytoplasm. Ini yang kita dah belajar hari tu. Jillian gives fresh frosty drink green tea pie green tea pie, orange topping truffle to Peppy P-E-P Popeye pyruvate. That's the 10 compounds we use in glycolysis. As I already emphasized the other day, make sure you understand and remember the name first. Ingat nama compound dulu, lepas tu dia punya proses akan jadi senang. So glycolysis dah from one molecule of glucose 6 carbon, dia jadi 2 molecules of pyruvate yang 3 carbon. Habis glycolysis bergantung kepada ada ke tak ada oksigen. Kalau ada oksigen, dia akan sambung link reaction or we call the pyruvate oxidation. Okay? During this link reaction or pyruvate oxidation, dia akan ada reaction that we call oxidative decarboxylation. Oxidative decarboxylation melibatkan oxidation, so ada oxidizing agent NAD+, and decarboxylation, delete carbon. So, dia dah hilang 1 carbon, dari 3 carbon, dia jadi 2 carbon sahaja. That's how from one pyruvate after link reaction or pyruvate oxidation, I akan dapat 1 acetyl-CoA. Make sure C dengan A huruf besar. And then, kalau 1 glucose, you get 2 pyruvate. That's why 2 pyruvate after link reaction, I will get 2 molecules of acetyl-CoA. That's at the end of link reaction. So far, so good.
Good, Miss.
Yes, Miss.
And when we say for link reaction, there will be no ATP produced. We only have NADH. 2 molecules NADH akan dihasilkan semasa link reaction. Whereas for glycolysis, through substrate level phosphorylation, I will be able to produce 4 molecules of ATP. Tapi sebab dah tolak 2 yang kita guna semasa energy investment phase, so I tinggal baki 2 sahaja. So 2 net ATP. That tells why only 2 ATP produced so far through substrate level phosphorylation semasa glycolysis. Link reaction tak ada. Tapi sini kita dah kumpulkan 2 ATP and then 2 NADH. Tambah dengan 2 NADH dari link reaction. Sini pun ada 2 NADH. Now, kita sambung dengan the third stage or the second main stage. Okay? Kita cakap Krebs cycle ialah stage utama yang kedua. Over here, 1 acetyl-CoA will enter the Krebs cycle, you get 1 turn. 1 acetyl-CoA, 1 pusing. But then, 1 glucose will give us 2 molecules of acetyl-CoA. That's why Krebs cycle akan pusing 2 kali. So 1 glucose molecule, there will be 2 turns of the Krebs cycle. Kalau 2 turns, kita akan dapat 2 ATP. And then, kalau 1 turn ada 3 NADH, so sekarang times 2 kita ada 6. And then FADH2 pun dekat step 6 ini, when succinate oxidize to fumarate, kita buat FADH2. So, dapat 1 FADH2 from 1 turn. Now, 2 turns, I will have 2 FADH2. Tambah dengan byproduct carbon dioxide from oxidative decarboxylation, kita dapat 4 carbon dioxide. Sebab ada 2 kali decarboxylation berlaku. So, So far, I get 2 ATP, 6 NADH, as well as 2 FADH2. And 4 ATP sahaja yang dihasilkan melalui substrate level phosphorylation. But the thing is, sepatutnya kalau ini ialah active cell. Contoh active cell ada apa? What are the examples of active cell?
Cardiac cell ka, Miss.
Very good. Cardiac cell and
Liver
what
cell.
more? Liver cell. Good job. So cardiac cell, liver cell, they are the active cell. They should produce 38 ATP in total. Dari satu glucose molecule sepatutnya akan dapat 38 ATP. Tapi sekarang semua hanya 4 sahaja. Mana yang lain masih dalam NADH and FADH2. We still have a lot of energy stored in NADH and FADH2 in the form of electron. Kita kena keluarkan energi dari elektron ini, dari NADH and FADH2. Itulah kenapa kita perlu sambung dengan last stage, which is oxidative phosphorylation. Oxidative phosphorylation, location dia dekat inner mitochondrial membrane or cristae. Pastikan you ada konsep yang betul lah. Inner mitochondrial membrane yang paling dalam ini. Make sure you remember ini ialah intermembrane space. Kita cakap inter sebab ialah antara membrane. Antara inner and outer membrane, so we call it intermembrane space. Yang dalam sekali inilah matrix, mitochondria matrix. Make sure you remember each part correctly. Okay. Inner mitochondrial membrane ialah lapisan membrane di mana oxidative phosphorylation berlaku. Ini ialah ruang, okay? Intermembrane space ini ialah ruang. Dia bukan satu lapis sel bilayer, bukan phospholipid bilayer. Inner mitochondrial membrane, yes, it is a bilayer, phospholipid bilayer. Sebab dia membrane. Matrix ini pun dia satu ruang. Ada fluid lah dekat matrix. So membrane is membrane. Space tak sama. So now oxidative phosphorylation, dia mesti start dengan electron transport chain dulu. Lepas tu baru chemiosmosis. And they must happen in series. Macam lepas ETC kena ada chemiosmosis. Alright. Now, tadi kita dah cakap semasa glycolysis, link reaction, Krebs cycle, dia akan produce NADH. And total, we have about 10 NADH produced. Other than that, we also have FADH2 produced during Krebs cycle. Ada 2 FADH2 yang produce semasa Krebs cycle. NADH dengan FADH2 ini ada high energy electron. Maksudnya kita nak keluarkan energi dari elektron ini. Apa yang kita boleh buat? Kita guna ETC. Nama pun dah lah, electron transport chain. Mestilah specifically for electron transport. And what do you need to remember on ETC? 4 complexes punya nama. NADH dehydrogenase. Complex 2 nama apa?
Succinate dehydrogenase.
Very good. Succinate dehydrogenase. And then complex 3, complex 4, nama dia hampir sama. You just have to remember redox. So, cytochrome C reductase dulu, baru cytochrome C oxidase. Other than that, we have 2 mobile electron carrier. Semua pun electron carrier sebab semua pun akan carry electron, terima and then pass the electron, accept and then pass the electron. Semua pun akan carry electrons. The difference is that four complexes ini tak boleh gerak. Tapi CoQ dengan cytochrome C, they can move. Sebab empat-empat ini tak boleh gerak, dia perlulah CoQ untuk tolong gerak pergi sini untuk ambil and then baru pass dekat complex 3. We need the mobile electron carrier to carry the electron from one complex to another. Try to imagine, kalau semua pun tak boleh gerak, macam mana dia nak pass electron? Tak dapat. So, kita mesti ada dua mobile electron carrier untuk tolong pass the electron between complexes. Make sense?
Yes, Miss.
Okay. Am I too
Yes.
fast? Boleh ya? Boleh faham ah?
Boleh, Miss.
Alright. Okay. So now we are going to see how this electron from NADH and FADH2 are taken out. Kita nak keluarkan elektron ini supaya energi tu kita boleh keluarkan. NADH akan bagi elektron, give elektron, transfer elektron pergi which complex?
Complex 1.
Very good. Nama pun dalam NADH dehydrogenase, maksudnya dia akan terima hydrogen and electron from the NADH. So, NADH elektron dia dah bagi dekat complex 1, so dia dah oxidize ke reduced? Dia lost electron. So, NADH
Oxygen.
very good. Oxidized to NAD+. So H dia pergi mana? H dia pergi dekat matrix lah sebab elektron je yang masuk. H2 pun masuklah dekat matrix. And now, this electron, will it go to complex 2 or CoQ?
CoQ.
Good job. It goes to CoQ. And then dari CoQ, dia pergi dekat
Complex 3.
complex 3, and then complex 3 pergi dekat cytochrome C, and then cytochrome C pergi dekat complex 4, and lastly, this electron will go to
Oxygen.
very good oxygen molecule, which is the final electron acceptor. That's how the electron from NADH ends up in the oxygen. Right, that's only for NADH. Selain daripada NADH, kita pun masih ada high energy electron dalam FADH2. So kita nak keluarkan electron from FADH2. So FADH2 dia bagi electron dekat which complex?
Complex 2.
2.
Very good. So 2, complex 2. So FADH2 pula tak bagi electron dekat complex 1, tapi dia akan transfer the electron to complex 2. And then dia sendiri pun oxidize jadi FAD. Samalah, H dia pun masuk dekat mitochondrial matrix. And then this electron from complex 2 will go to
CoQ.
CoQ. And the rest, they are the same. Just the beginning je yang tak sama. NADH transfer electron to NADH dehydrogenase. Tapi FADH2 transfer the electron to succinate dehydrogenase. Ini je yang tak sama. Lepas dua-dua dah bagi elektron dekat CoQ, yang seterusnya semua sama. CoQ to complex 3, which is cytochrome C reductase, and then pergi cytochrome C, and then cytochrome C oxidase complex 4. Barulah dia sampai oxygen molecule, which is the last electron acceptor. And kita nak balance equation. That's why this is half. And that's how we get water, which Jordan asked yesterday. This is the byproduct of cellular respiration. Dari mana dia dapat H plus ini? Memang dalam mitochondrial matrix dia ada H plus. That's all about electron transport chain. From beginning, lastly, masuk dekat oxygen molecule combine with H plus to form water. Any question?
No, miss.
So far, we have only formed the water, okay? Tapi yang kita nak buat ialah ATP. That's why if we just stop at ETC, the production of ATP is not complete yet. Kita belum produce ATP kalau kita hanya ada electron transport chain. So how? We must coordinate with chemiosmosis. Dia kena berlaku juga chemiosmosis supaya dia dapat produce ATP. Kenapa kita namakan ini oxidative phosphorylation? Sebab cara kita produce ATP ini melibatkan redox reaction. That's the reason why we call the name as oxidative phosphorylation. So now, during redox reaction, dia akan ada electron transport. When the electron transport akan ada energy release. What does this energy release during electron transport used for?
To pump the hydrogen from mitochondrial matrix to intermembrane space.
Very good. To pump the hydrogen, which is the protons, from mitochondrial matrix to the intermembrane space. And kalau you perasan, cara yang Miss lukis memang complex 1, 3, and 4 sahaja yang akan pump the hydrogen from the mitochondrial matrix to the intermembrane space. Complex 2 tak terlibat. Hanya 1, 3, and 4 je yang terlibat dalam pumping the protons from the mitochondrial matrix to the intermembrane space. And this will cause the concentration of hydrogen ions or protons to be high or low dekat intermembrane space.
Hi.
Hi.
Very good. High. Whereas for mitochondrial matrix pula sebab hydrogen ions, protons dah keluar. So dia dah jadi low concentration of H+. Good job. So far, so good.
Good, miss.
And like what I told you yesterday, we must first create the difference in concentration. Okay? Mesti buatkan ada perbezaan concentration of protons dulu dekat intermembrane space and mitochondrial matrix. Kalau dia dah ada different concentration, barulah dia akan jadi ada satu force, the proton motive force. Dan force ini dia buat apa? Force ini untuk force the hydrogen ions or the protons back to where?
Mitochondrial matrix.
Very good. Back to the mitochondrial matrix sebab dia nak balance the concentration. So back to mitochondrial matrix, this is down the concentration gradient. Sebab from high to low. So kita cakap down the concentration gradient. Mesti dari high dia balik low. And what for? To balance the concentration of protons on both sides. But the problem now, inner mitochondrial membrane or the crystal, it is impermeable to H+. So kalau protons nak balik matrix, macam mana dia nak balik? Semua ini tak telap, tak boleh lalu. Dia hanya boleh lalu dekat.
ATP synthase.
ATP synthase.
Very good. ATP synthase. Okay, so dia dah balik. Dah lalu dekat ATP synthase untuk balik ke mitochondrial matrix so that dia boleh balance the concentration on both sides. So dia balik. Now I will show you a video on how the flow of protons activate the ATP synthase. This is it. Tak ada bunyi ya, tapi yang blue dots ini ialah hydrogen ions. So you can see these hydrogen ions will bind to this rotor. Okay, dia masuk and then dia bind, dia akan rotate rotor ini. And when this rotor rotates, dia akan rotate the knob underneath. So dia tadi bagi tahu, one hydrogen masuk, satu turn. One hydrogen masuk, satu turn. So when it turns, dia akan move this knob dekat bawah. So what for? Knob inilah yang dia akan buat ATP. So dekat knob ini, they will have ATP production. From ADP and inorganic phosphate, they join and become ATP. That's how the whole flow of hydrogen ions move the ATP synthase to produce ATP. This is how they do it. So dia mesti start dengan ada hydrogen flow, barulah ATP synthase ini dapat buat ATP. Jelas?
Jelas, Miss.
So that's how they do it. And that's the reason why kita hanya bagi laluan dekat ATP synthase sahaja. So that semasa semasa hydrogen ions ini move down the concentration gradient, dia move dari high concentration to low concentration melalui ATP synthase, dia boleh rotate the ATP synthase. And when it rotates the ATP synthase, that's when they will make ATP from ADP and inorganic phosphate. That's it. This is how we want the production of ATP using redox reaction. And that's why we name it as oxidative phosphorylation. So far, so good?
Good, Miss.
Yang lain semua faham, ah? Yeah?
Yes, Miss.
So what else do you need to know? One NADH will give you 3 ATP, whereas one FADH2 will give you 2. And I was just notified today that actually dia bukan ngam-ngam 3 ATP. This is extra information, okay? Extra information to tell you is that actually one NADH just give you 2.5 ATP, whereas one FADH2 it just give you 1.5 ATP. Tapi untuk senangkan student buat calculation, kita round up. So jadi 3 ATP dengan 2 ATP. If you use 2.5 ATP times 10, I only get 25 ATP. And then 1.5 times 2, I only get 3 ATP. So total ATP tu akan jadi kurang lah. Memang tak salah kalau you baca dekat mana-mana buku yang dia cakap, oh total ATP hanya 35. That's right too. Because from the way that 2.5 ATP je, we only get 25 from here, and then 3, so 32 ATP. Dia macam 28 tambah dengan 4 dari substrate level, we only get about 32 ATP. Ini memang tak salah sebab dia ambil satu NADH equal 2.5. Tapi ikut syllabus lah, kita ikut syllabus. Kalau ikut syllabus, dia nak senangkan pelajar, dia memang bagi round up. One NADH give 3 ATP, so one FADH2 give 2. Untuk senangkan you all calculate and remember. So ini hanya Miss nak share je. So in the exam, you follow this one. Did I make it clear?
Yes, Miss.
Yes, Miss.
Okay. So ini memang tak salah lah. Kita nak lagi spesifik, memang macam tu. Tapi untuk senangkan kita kira kita round up. So jadi 1 NADH, 3 ATP produced during oxidative phosphorylation, whereas FADH2, it will produce 2 ATP during oxidative phosphorylation. And now I'm going to tell you why FADH2 has lower ATP production. Kenapa NADH boleh dapat 3? Kenapa FADH2 hanya 2? This is extra information too, and it's in your notes. I ada bagi dalam nota sebagai extra information on page 8, slide 4. I ada label not important, this slide. Kalau you tengok nota muka surat 8, slide 4, adalah slide ini. And why do I include this is just to let you know a bit more. Why FADH2 can only produce 2 ATP or 1.5. Why is it less than NADH? This has something to do with the complexes. NADH dia transfer electron pergi complex 1. FADH2 transfer electron pergi complex 2. When we compare the energy of these complexes, kita bandingkan dia punya potential energy. Ini ialah complex 1. Ini ialah complex 2. Okay? We can see that the energy level for complex 1 is higher sebab dia semakin turun, semakin kurang potential energy. You can see high energy to low energy. So apparently NADH bagi electron dekat complex 1 is higher energy. Whereas FADH2 bagi electron to complex 2 is lower energy. And that tells why FADH2 hanya dapat produce 2 ATP during oxidative phosphorylation. Jelas?
Jelas, Miss.
Jelas, Miss.
Okay, so ini hanya extra information. You just have to remember that as the complexes go along the complexes, the energy release tu dia progressively lower energy level. So NADH dekat complex 1 ini yang highest energy lah, sebab tu dia dapat produce more ATP. Sebab dia bagi electron dekat complex 1 with higher energy level. Whereas complex 2 lower energy level, so FADH2 dia start dekat complex 2, mestilah dia pun dapat produce less ATP, only 2 in this case. That's all I want to share with you about the production of ATP. So itu je pasal oxidative phosphorylation. I hope by now you are clearer about the concept and ATP synthase. Any question that you would want to ask?
No, Miss.
No?
No, Miss.
Alright, so make sure you tahu maksud proton gradient sama dengan proton motive force. Sama je maksud concentration difference. Okay, good. Alright, so tu je pasal aerobic respiration. Now let's look at the questions together. So ini soalan 4. Okay. Figure 4 shows the process of cellular respiration in liver cells. Okay, liver cells, is it active or normal cell?
Active.
Very good. So ini ialah active cells. Okay. Kalau active cell, soalan tak tanyalah sekarang, tapi kalau active cell, how many total ATP are produced during aerobic respiration?
38.
Very good. 38 ATP. Okay, kita masih ikut syllabus 38. Tapi you jangan rasa confused. Kenapa ada buku tulis 32? Itu memang ikut yang Miss explain tadi. But we follow the syllabus. For now, we still say that the total ATP is 38. I just want you to know that it's not wrong. If you read somewhere, dia cakap 32. And I don't want you to confuse, so for the time being, I will still teach based on 38 ATP. So define aerobic respiration. Ini boleh ikut definition yang Miss bagi dalam nota. Okay, so the complete oxidation of glucose into carbon dioxide, water, and ATP or energy in the presence of oxygen. Itulah definition dia. B. How many ATPs are produced from substrate level phosphorylation from one molecule of glucose in Krebs cycle? How many? Substrate level je ya.
2.
2. Good job. 2 je. And then C, kita dah bincangkan. Now D. Cytochromes are parts of electron transport chain. Name the class of enzymes that can be found in the ETC. Right, kita dah belajar semalam. ETC always about electron, so always about redox. So what is the class of enzyme involved?
Oxidoreductase.
Good job. Oxidoreductases. So the next one, what will be produced when an electron is transported through the chain? Dia nak products from oxidative phosphorylation. Ini dia punya cara, I tak suka soalan dia sebenarnya. Sebab kalau what will be produced when an electron is transported through the chain, I feel like it's not complete. The question is not complete, but the way they ask this is to know the products of oxidative phosphorylation. So can you tell me the products of oxidative phosphorylation? Apa yang akan produce?
Carbon dioxide.
Carbon dioxide during oxidative? Are you sure?
ATP and water.
ATP and
Water.
water, that's it. Aduh, mana ada decarboxylation? Ingat ya, kalau decarboxylation barulah ada carbon dioxide. Tapi kalau oxidative phosphorylation, rujuk balik yang kita belajar ini, dia produce water dengan ATP sahaja. Itulah kenapa Miss cakap I tak suka soalan ini sebab what will be produced when electron is transport? I feel like it's just water. Kalau dia tak cakap dia ada chemiosmosis. But if it says and continue with and follow by chemiosmosis, then I will consider ATP. Tapi sebab soalan memang 2 markah, so kenalah kita jawab juga ATP. Kena pandai-pandailah ikut dia punya markah. Kalau 2 markah, then you should consider ATP as well. Sebab kalau electron transport chain sahaja, dia memang hanya produce water. Okay, so 2, water and ATP.
Okay,
Can we
Miss.
move on? Okay, move on to the next question. This one, kita tak belajar E. Semalam kita tak bincang yang ni. Figure 3 shows some of the stages in the Krebs cycle, and now E dia tanya, hydrogen from the Krebs cycle enters the electron transport chain and oxidative phosphorylation occurs. See, it's very complete. Lepas ETC, dia cakap, okay, so semasa ETC ini, dia ada oxidative phosphorylation occurs, and they ask you to explain what is meant by oxidative phosphorylation. Kalau substrate level phosphorylation memang dah pernah keluar dalam past year, and Miss dah bagi definition dia dekat last page of your notes. Page yang terakhir, slide yang terakhir. Memang ada the term substrate level phosphorylation meaning. But now, tak pernah keluar dalam exam pasal oxidative. What if they ask you about oxidative and 3 marks? So inilah cara you explain. Okay? First, bagi tahu dulu, electrons tu akan pass along the ETC. Sebab dia dah cakap, hydrogen from Krebs cycle enters ETC. So ETC always relate to electron. electrons from the hydrogen atoms are passed along the ETC and always end with the final electron acceptor, which is oxygen. Sebab dia nak tahu oxidative phosphorylation, so kita bagi tahu redox dulu. Melalui redox reaction produce ATP. So bagi tahu dulu redox reaction berlaku dekat mana, dari apa sampai mana. So electrons along the ETC until oxygen, which is the final electron acceptor. Done. Ini dah terangkan ada redox reaction, ada oxidative part. But then, what about phosphorylation part? Ini semasa chemiosmosis. As the electrons move along the electron transport chain or as the electrons move during redox reaction, energy is released. What for? The energy released is used to create the proton gradient. Okay? It's used to create the proton gradient, which then used to phosphorylate ADP to form ATP. Ini, sorry ya, an inorganic phosphate. Ini je, 3 markah. You will summarize all the points in ETC and chemiosmosis into 3 marks. First, jawab dulu apa itu oxidative. So terangkan ada redox reaction semasa ETC. That's what we say here. And what about phosphorylation part? You kena bagi tahu dulu apa berlaku semasa phosphorylation untuk buat ATP, dan you kaitkan redox reaction ini buat apa, redox reaction ini buat energy released, which is used to create the difference in concentration of protons, the proton gradient. Dah ada proton gradient, barulah dia boleh tolong untuk form ATP from ADP and inorganic phosphate. Ngam-ngam 3 markah. Okay?
Okay, Miss.
Right. Boleh catat yang ni. You boleh ambil gambar ke atau you nak salin? So boleh salin. The whole concept is like what I terangkan panjang lebar tadi. You just summarize everything into 3 sentences, and that tells what is oxidative phosphorylation. Hopefully, if it comes out in the exam, you will be able to explain it in short and detailed way. So that's it. Alright. Dah habis salin atau do you need time to copy?
Sudah, Miss.
Sudah?
Sudah, Miss.
Alright. Okay, so that's it for this question. And now the next one yang Miss suruh you cuba buat, kita bincanglah soalan ini dengan jawapan dia. Dalam past year, kalau you perasan, tak pernah pun keluar ETC dalam, tak pernah keluar oxidative phosphorylation dalam structure. What if they have it in structured question? So I just give you this one as an example. Biasanya dia keluar dalam essay, tapi mana tahu kalau dia keluar dalam structure, can you answer it? So let's try. First, define redox reaction. So redox ada apa dengan apa?
Reduction dengan oxidation.
Yeah, itu je. Okay, it's just reduction and oxidation, or if you want to say gain and loss electrons, itu pun boleh. Receive and loss of electron or gain and loss of electron, I pun terima. Tapi kalau I tulis oxidation and reduction lah. And then B, name complexes 1, 2, 3, and 4. That's the reason why I ask you to memorize the name. You memang takkan ada problem memorize NADH dehydrogenase. Tapi ramai student akan silap untuk complex 2. Please remember it is succinate dehydrogenase. Jangan pergi tulis FADH2 dehydrogenase. Salah. Succinate dehydrogenase. Dia ikut step Krebs cycle where FADH2 is produced. And then complex 3 ialah cytochrome C, reductase ke oxidase?
Reductase.
reductase. And then complex 4 ialah oxidase. Cytochrome C, oxidase. Tu je. So this is succinate dehydrogenase. Itulah dehydrogenase, maksudnya dia delete hydrogen. Delete hydrogen atom. So when the hydrogen atom is delete, H plus dalam matrix, electron masuk dalam complex. C. State the process that occurs at Q and K respectively. Q ialah
Electron transport chain.
ETC.
ETC, electron transport chain. Write in full will be better. Okay. Even though sometimes dia memang tak penalize, dia memang terima juga ETC, tapi lagi selamat tulis electron transport chain. And then for K, this will be
Osmosis.
chemiosmosis. Chemiosmosis. Okay. Now D. D dengan E, put a star there because it's a good question. Tak dapat letak star. What would happen when a poison inhibits complex 4 at Q? Complex 4. Kalau you masih ingat poison tu, poison apa tu?
Carbon monoxide ka, miss.
No. Carbon monoxide tu dekat blood. Carbon monoxide you will learn later when we learn gaseous exchange, but not here. What is that? The poison that will affect complex 4. Start with C juga. Cyanide.
Cyanide.
Yeah. Class, imagine in our food, we do have cyanide. Where? Apple seeds. Sem 1 masa Miss ajar apple, bukan ajar pasal apple. Ajar flowering plant, I did talk about cyanide in the apple seeds. So we try not to eat the apple seeds when you eat the apple because inside the apple seeds it contains cyanide. So if you consume a large amount, bukan makan satu dua you akan mati. But if it's a large amount of apple seeds, it will give you a large amount of cyanide that can cause death. How? This is how. Cyanide is the type of poison that can inhibit or block complex 4. And like yesterday when I taught you, if complex 4 dia dah tak boleh function, what will happen then? Complex 4 dah tak ada. Apa? Boleh form water?
Tak boleh.
Tak boleh.
No.
ETC ini complete ke? Tak sampai akhir pun. Is it complete?
No.
No, miss.
That's it. So it means that the electron transport chain is stopped and oxygen pun tak boleh jadi water. I don't like this equation. Ikut balik ya yang Miss bagi tu. Jangan ikut yang ni. Ikut yang Miss bagi ini sebagai equation dia. Okay? So this is the reason. What would happen? Terus ingat, okay, complex 4 dah tak ada. Maksudnya the whole ETC dah tak complete. It means the ETC is inhibited. The transport of electrons along the chain is blocked, inhibited. Itu one mark. And then try to relate. Complex 4 dia terlibat dalam apa? Dia terlibat dalam bagi elektron dekat oksigen. So it means that if without complex 4, electron cannot be given, tak boleh reach oxygen, so water will not be formed. Oxygen is not reduced to water. Make sense? Point 1, 2 ini boleh faham?
Boleh, miss.
Okay. So apparently kita dah kurang satu kompleks untuk pump hydrogen. Tak boleh cakap, oh ATP tu kurang kot sebab hydrogen dia kurang. Padahal dia akan buatkan the whole ATP synthesis stop. It's not just become less. It may be so inefficient that sampai dia stop terus the ATP production. This is what the answer given, but I will have to confirm again whether they produce less ATP or whether they stop terus. But so far yang Miss belajar dia stop terus lah. Next week I will get back to you, okay? Whether they will produce a fewer amount or whether dia memang unable to produce. Sekarang ingat yang dua ni dululah, redox reaction block, and then water cannot be formed. Are you okay?
Okay, Miss.
Okay.
This is a very good question and it may come out in the exam. So far tak pernah keluar lagi, tak tahu mungkin akan keluar. And now the last question here. Explain the link between the formation of ATP and the hydrogen ions shown at K. Actually, this is another way of asking oxidative phosphorylation, tapi dia nak you bagi tahu juga pasal H+, sebab dia dah specify H+ dekat sini. So you have to tell what is the role of H+ here, what happens to the H+ concentration, and how does that give rise to ATP production. First of all, how can H+ give rise to ATP production? Macam mana?
Proton gradient.
Okay, I would start with proton gradient even though dekat sini jawapan dia tak cakap. Tapi I will start with the difference in concentration in the intermembrane space and mitochondrial matrix. Okay, I will start with that. So proton gradient is created due to the difference in concentration of H+ or protons in intermembrane space and mitochondrial matrix. I akan tambahlah point ini. Okay, tak tahu dia perlu ke tak, tapi I tambah je because it's related to H+. So proton gradient is created due to the difference in concentration of H+ in intermembrane space and mitochondrial matrix. So the next one, it will be protons buat apa? Or H+.
Down the concentration and activate the ATP synthase.
But you have to tell through what? Get down the concentration gradient through what?
ATP synthase.
Very
ATP
good.
synthase.
Yes, good job. So kita cakap protons diffuse through ATP synthase. Okay, kena bagi tahu dulu. Proton diffuse through ATP synthase down its concentration gradient. Okay, through ATP synthase down its concentration gradient. And if you want to be more specific, you boleh bagi tahu from intermembrane space to mitochondrial matrix. Ini yang student selalu salah. Selalu salah tempat. They will say it differently. Please remember it's between intermembrane space dengan matrix. So dari intermembrane space, the H+, it will diffuse down its concentration gradient, dari high concentration to low concentration through ATP synthase. And what happened then? During the flow, what will be released? Semasa dia flow, akan jadi apa? Apa akan jadi? Akan jadi apa pula? Apa akan jadi? ATP synthase dia akan
Activated.
tapi activated maksudnya dia uses ATP synthase uses energy from where? Apa yang bagi energy ini? Dia activated sebab apa? Dia activated sebab apa yang buatkan ATP synthase activate?
Proton passes through.
Good job. Protons passes through. And you should remember that, like I showed you the video just now, when the H plus flow through, actually it gives out some energy. Energy inilah yang diguna oleh ATP synthase untuk buat ATP. So we say ATP synthase uses energy from the flow of H plus. You say like this, tak boleh hanya cakap activated. Macam mana dia activated? It's because the flow of H plus, semasa H plus go through the ATP synthase, dia akan release energy. Dan energy inilah yang digunakan oleh ATP synthase to catalyze, okay? To catalyze the formation of ATP from ADP and inorganic phosphate. Okay, tak boleh hanya cakap activated ya. So always have to mention the flow of H plus. The flow of H plus ini penting untuk ATP synthase. Sebab masa dia flow through yang dia ada energy, and energy itulah yang kita gunalah untuk form ATP from ADP and inorganic phosphate. Itu je. That show you how H plus is related to ATP production. Jelas?
Jelas, Miss.
Okay, boleh jawab ya?
Boleh.
Hopefully. Oh well. Alright, so any question on this?
Miss,
Yeah.
yang cyanide tadi tu, dia memang inhibit complex 4 saja ke? Dia tak boleh complex lain ke?
Dia hanya add on complex 4 saja. It's like macam kita cakap competitive inhibitor pun, when we talk about malonate, ingat lagi ke malonate dengan succinate? Succinate itu pun dia hanya target dekat enzim apa dah? Succinate dehydrogenase. Bukan kan? hold on. I dah tak ingat dia enzim apa tu. Wait. Succinate dehydrogenase. Betul lah, succinate dehydrogenase itu pun hanya dia add on succinate dehydrogenase je as the competitive inhibitor. So sama lah, dia bukan succinate dehydrogenase, dia tak sama dengan yang ni. And they will not target other enzyme, they will only target certain type of enzyme je. Cyanide, it only targets the complex 4, cytochrome C oxidase sahaja. Tapi dia hanya mempengaruhi satu kompleks, itu pun dah cukup untuk affect the whole ETC. Okay.
Okay, Miss. Thank you.
Alright, thank you for asking. So other than that, is there any other questions on oxidative phosphorylation? Alright.
No, Miss.
No, then kita take 5, and then I will continue with fermentation. So I will come back on 15:10. So 3:10.